What's going on, my friends? Welcome back. It's Whiteboard Wednesday, and so today we're going to talk about a really heavy topic: how autoimmune disease unfolds. I decided that this was something that was worth discussing because it'll help us get a sense of how autoimmune disease obviously manifests, and understanding that we can also reverse this because we can see the mechanisms that are taking place and the things that are triggering the autoimmune attack. Okay. Now, before I begin, we should understand too that there are 50 million Americans that have autoimmune disease, okay? And the annual rates are increasing. So somewhere between 2 to 7%, I think, depending on the disease. So it is pretty significant, right? Now, we need to understand that 30% of autoimmune disease is related to genetics, okay? It's mostly environment. So things like your diet, pathogens, toxicants, metals, things like that. I think we know that food is a very obvious trigger, right? Some foods are more immunogenic than others. So wheat and dairy in particular are very immunogenic in people with autoimmune disease compared to other foods. And even that is very disparate. So some people are going to be more reactive to certain foods like soy and nightshades compared to others who maybe aren't. Nonetheless, these foods are capable of cross-reacting with human tissue, like human islet cells, myelin causing MS, your thyroid causing Hashimoto's, aquaporin in your eyeball causing neuromyelitis optica. Okay, so these are some of the outcomes from food and immune cells interacting. We also have lots of emerging research that shows that pathogens, viruses, bacteria, mold, fungus are all capable of inducing autoimmune disease. Okay, so Epstein-Barr, measles, Lyme disease, all strongly implicated in multiple sclerosis, West Nile virus, and myasthenia gravis, streptococcus and pandas, right? Cytomegalovirus, Epstein-Barr and Hashimoto's, P. gingivalis and rheumatoid arthritis, and I can go on and on and on. You get the idea. So these have a profound impact on predisposing us to autoimmune disease. And then you have this huge list of toxins, right? Thousands of them out there. BPA, heavy metals, mercury, aluminum, arsenic, right? Tetrachloroethylene and dry cleaning, formaldehyde. I've even seen research on nail polish causing autoimmune disease. It's crazy. So here's kind of what happens actually. So when you have a cell, right, you get something like mercury that enters the cell. It's going to covalently bind with your human tissue, and then it's going to release the cell, and you have this new thing that's called a neoantigen. It's 50% your own tissue, 50% this metal, right? And then your immune system's like, what the hell is this, right? So you have this macrophage that's going to come and bind to it, and it's going to say, hey, T cell, check this out. I got this thing here. T cell binds to it, picks it up, and then it signals your B cells, right? And they basically differentiate into plasma cells, and they make antibodies. So now you have antibodies being made against this thing, right? This mercury, this compound here, and against your own tissue, because it sees this thing and it's like, okay, well, it's 50% my own human tissue, right? So I'm gonna flag that and stimulate T cells to attack it. Now, before I get into the different immunological mechanisms that kind of underlie autoimmune disease, I should also mention that these chemicals are capable of causing epigenetic changes, right, at the DNA. So they change the expression of the DNA that are coded for making proteins. So maybe there's a dysfunction in a protein that's responsible for keeping your gut intact or making an immune cell, right? These toxins, these pathogens can cause changes that are going to really put you at a disadvantage. So the first thing we're looking at is molecular mimicry, which is a super common occurrence in autoimmune disease, right? So if we look at gluten and how that contributes to Hashimoto's or celiac disease, it's because of this cross-reactivity that's going on here. So say, for example, this is like a gliadin molecule or a bacteria, right? So it gets sucked up by this macrophage, and it's going to present it on its toll-like receptors here, which are specific for certain microbial antigens, or epitopes rather. Epitope is the part of the antigen that the T cell, which is this thing right here, is going to bind to, okay? So this phenomenon is where the T cell binds to this epitope, part of the bacteria, for example, and it then cross-reacts and attacks your own tissue, like your thyroid or your gut lining, right, and celiac, because it looks identical to your own tissue. Now, what can also happen too is that if you have your own tissue, say right here, your own cells, these bacteria, as an example here, can infiltrate go into the cell, right? And it can come out, and, well, rather, what it does is it kicks out these self-antigens, these self-proteins, right? So these things kind of get released because the bacteria is destroying it, right? And they get sucked up into the macrophage, right? And so that gets mixed up with this bacteria antigen too. And like we talked about with the heavy metals in the other clip, we can see that this same thing can happen where it's going to look at the bacteria in your own tissue, see that it's got both things, and then therefore attack your own tissue because it looks the same. Epitope spreading is like when a virus invades a cell. It manufactures its own proteins for its own survival. And then the cell ruptures, it goes through lysis, and the proteins get out. There's tons of them. And then it alarms the immune system, the antibodies come to tag it. And that can induce the molecular mimicry phenomenon as well. Now, the other thing that can happen is when you have your macrophage, right, it's taking up this bacteria like before, right? Now, the bacteria simultaneously could be hitting your cells, you know, and then causing the release of your own tissue beneath it, right? These little antigens, and they get sucked up into the macrophage. And then suddenly you have your T cells that are going to react and bind to that, right? They're going to bind to, well, not only the bacteria antigen, but also your own antigen, right? Now, when that happens, you have the release of pro-inflammatory cytokines and mediators and things like that, which damage your own tissue even more. And then you have the release of more of your own self-antigens, right? And so these look different than these guys, and they're called super antigens. And then they get sucked into the macrophage, and they get presented to the T cells, and now they're seeing a whole new molecule. And this phenomenon keeps going on and on. Those T cells are going to release more inflammatory cytokines and do more damage, and so you have this spreading of more and more new epitopes, right? New molecules that were not there before, right? That's how that happens. And this phenomenon keeps going on and on. Those T cells are going to release more inflammatory cytokines and do more damage, and so you have this spreading of more and more new epitopes, right? New molecules that were not there before, right? That's how that happens. So before we move on, since we're talking about bacteria, right? In rheumatoid arthritis, there is a bacteria called P. gingivalis, causes gingivitis, as you might assume. And this guy makes an enzyme called PAD4. It's peptidyl arginine deaminase, I think, and it deaminates this amino acid arginine. It adds water to it, and it rips off NH3, and it becomes citrulline. This all of a sudden becomes a molecule that the immune system has never seen before, and it's like, what the hell is that? So it's making antibodies and T cells against that. That's why when you check for rheumatoid arthritis antibodies, you're not only doing rheumatoid factor, but anti-CCP, right? Cyclic citrullonated peptide. That's what that is. You're checking for antibodies against that. This is not correlational data. This bacteria can cause rheumatoid arthritis. It is not the only thing that can cause it, but it's one of these super underrecognized and missed factors. Another quick example is rheumatic fever. So in this autoimmune disease, you have streptococcus, this bacteria that's making streptococcal M proteins and other antigens that look like your heart valves, your joints, your brain, your collagen, right? So your antibodies are attacking the bacteria and like, hey, that looks a lot like these other areas of my body. All right, now lastly, we have the bystander effect. So kind of like the other ones, you have your human cells there, you have the bacteria up here, it enters the thing, the macrophage, and simultaneously it's entering your own tissue and it's causing the release of these self proteins. They get sucked up into this macrophage, and so then you have your T cells that are binding to not only your bacteria, right, but also binding to your own proteins right here, right? Now, in this entire process, what happens is that these T cells release these pro-inflammatory cytokines and mediators, like I talked about before, and they cause more damage, right? And they release more self-antigens. Okay, this is the bystander effect. This is essentially what that is, right? It means that the tissue becomes a bystander in the immune battle against the infection, in that local space. Okay, so T cells don't have control over their activity, right? They're trying to handle this bacteria, but they're inadvertently destroying tissue everywhere in that vicinity. So that's it. That's what's going on with autoimmune disease. That's pretty much as far as we know. So as you can see, toxins, heavy metals, infections, viruses, bacteria, mycotoxins, Lyme, food, all these things can trigger these sort of mechanisms and cause autoimmune disease in people who are genetically predisposed. So that's it. Thanks for tuning in. See you next time.